Good morning, or good afternoon, or good evening, depending where you are and when you're listening to this. And all our live attendees are very welcome. And you're also very welcome if you're watching the recording of this on the FEMS YouTube channel or on another link. My name is John Morrissey. I'm the Editor-in-Chief of FEMS Yeast Research, and I'm your host for the webinar today, which is entitled Advances in Synthetic Biology Tools to Engineer Yeast Cells for Biotechnology. And our speakers, as you will know, are Hal Alper and Jin Hao. Just before we get into the substance of the webinar, I just want to say a little bit about FEMS, which is the Microbiology Society, which is hosting today's webinar in conjunction with our publishers, Oxford University Press. So FEMS is a federation of European microbiology societies. And in fact, this is a collection of more than 50 mainly national member societies, which cover all aspects of microbiology. And we have over 30,000 members between all of these different European microbiology societies, covering all disciplines, all sub-disciplines of microbiology, fundamental research to application, teaching to research, clinical practice, and other types of practice as well. Now, the aims of FEMS as an organization are really to invest back into science and to build communities of our researchers. And to do that, we run a range of different kinds of activities, including conferences, workshops, grant schemes, and publishing. The Society publishes 7 scientific journals, 2 of which are very new, MicroLife and FEMS Microbes, and these were launched in the last year. And then we have 5 journals which have been on the go for quite a few years. And you can see these listed across your screen here. The most important of these journals is FEMS Yeast Research, of course. And these journals are, the latest 2 are fully open access journals, whereas the first 5 are hybrid journals. And so you can publish for free in the subscription type of model, or you can publish fully open access as well. So we will publish in any of those styles of papers. Today's webinar, as I said, it's hosted by FEMS Yeast Research. And this is actually the first in what we hope will be a series of webinars. These webinars have been run by FEMS and OUP over the last few months for some of our other journals like FEMS Microbiology Ecology, and they've been very successful with a lot of attendees. So I'm pleased to see there's over 200 people watching here at the moment. We will have more webinars following on from this one on different topics. The journal itself, it's a broad scope yeast journal. It's really a journal of the yeast community where, because FEMS is a learned society, resources and funding that are generated by the journal go back into the scientific community to fund those different activities I mentioned earlier, like grants and conferences. I'm the editor-in-chief of the journal. The deputy editor-in-chief is Dr. Carl Munro in Aberdeen. We're a broad scope journal, so really our aim is to publish good research from any aspect of yeast, so fundamental genetics and molecular biology, right up to industrial biotechnology, Saccharomyces and non-Saccharomyces, physiology and metabolism, systems and synthetic biology. So we really, any aspect of yeast research that's of interest to the yeast community falls within the scope of our journal. Now, to help with the publishing, we have a very international panel of editors who you can see on your screen there, covering most aspects of the globe. So even though FEMS Yeast Research is it's a journal of FEMS and published by OUP, it's a global publication, and this is reflected in our editorial board. Our editors who you see on top here. And we also then have a very strong international editorial board that's spread right across the globe. And this editorial team is actually really important because they ensure that we maintain the high quality of standards for the journal. And it's one of the things that we will say about the journal. It's a journal for the yeast community where we publish articles from any area of yeast research, but we have high standards of peer review and assessment to make sure that the quality of the research that's published in any of those areas is high. And I would say you can rely on FEMS Yeast Research only to publish quality research. In addition to primary research articles, which are published as they are accepted, the journal also publishes regular thematic issues on important areas of yeast research, or on topical issues related to yeast research. The most recent was one on yeast synthetic biology, which is linked to today's webinar. We also had an anniversary thematic issue earlier this year, and last year, the last one of last year was on ecology. But you can go onto the FEMS Yeast Research website and you can see these different thematic issues. We've got a really exciting portfolio of thematic issues coming up for 2021 and 2022 as well. Some of those are almost completed and some are in progress. And that really brings me to the end of my introduction and the start of the substance of today's webinar. And I'm really pleased to have the two speakers that we have, Hal Alper from the University of Texas at Austin and Jin Hao from Shandong University in China, who are going to give talks on the titles you see there, biosensors and CRISPR-Cas. These two speakers publish articles in that recent thematic issue I mentioned on synthetic biology for biotechnology. Indeed, Jin Hao was one of the editors of that thematic issue. I'm not going to say anything about really much about the importance of either synthetic biology or biotechnology, because I think the fact that you are listening here, you appreciate the significance of this area. It's clear that synthetic biology has been revolutionized in the last 5 or 6 years by the introduction of new technologies. And I'm sure that Hal is going to talk about that. And these technologies are being applied in the area of metabolic engineering to engineer yeast for biotechnology applications, amongst other things. And despite the fact that we've got great tools now, there's still a lot of challenges in this area. And I'm really looking forward to Jin's presentation on biosensors, because this will maybe help highlight some of the emerging areas in the field of metabolic engineering. So with that now, I would like to say thank you to everyone who's in attendance here. Thank you to my speakers. In terms of the format, Hal will give his presentation for about 20 minutes. Jin will give her presentation for about 20 minutes, and we will have a Q&A after that. But you may submit your questions at any time. I think there should be a panel on your right where you have the opportunity to write questions. So you can submit your questions. They won't actually be seen by the speakers in real time. I'll be monitoring the questions, and I will put your questions to the relevant speaker in the Q&A after both of the presentations. So I think that that covers me, and I'll now pass over to Hal for his opening talk.
Thank you very much for that great introduction and overview to this series. It's a great opportunity to be able to present our work, obviously within this confines of a virtual world, but I think that these types of seminar series and symposiums are quite helpful for being able to disseminate information. So appreciate everyone who was able to join in for this. And as John mentioned, I'm here today to talk a little bit about some of the topics that had been relevant in our review article, but mostly in the discussion of how CRISPR-based technologies in particular have been able to be used not just for metabolic engineering, but for newfound ways to do functional genomics and general understanding of gene function. And needless to say, obviously the CRISPR revolution is here, and we have a new set of tools, and these tools can be used for genome editing, but also for gene expression control. And And really the topic I want to talk about today is the way in which we can use CRISPR-based systems for the control of gene expression. And in particular, by using deactivated Cas9, so dCas9 type systems, either on their own or linked with activators or repressors, we can actually begin to create a rapidly deployable tool set to be able to reprogram gene expression. And what I mean by this is that we can actually take these constructs, create them, have them in the freezer, and then even port them later on into new strains of interest. So it allows us to very quickly, rapidly prototype gene expression patterning that we want within a given cell. And this is was the dream to some extent of recombinant DNA technologies for quite a while. So we're really excited to be able to have these tools at our disposal. And it really brings up the question, if we can engineer the expression of every gene, and if we can tune the expression of every gene, what do we do with that? What do we do with that level of information? How do we go about taking this higher dimension of understanding of gene expression and map that back to metabolic pathways, to functional annotation, to the function of unknown genes? So this is really the promise of that overall field. So I'm going to focus a lot on metabolic engineering applications in terms of how we can identify gene targets using these types of approaches. So we started some of this work from our lab a couple of years ago in developing a technique that we call STEPS, which is systematic testing of enzyme perturbation sensitivities. And really what we use, our basic premise, is that we can use this idea of CRISPR-Cas9 based actuation of gene expression and look at that singly or in a multiplex fashion across pathway enzymes. And this is a way really to be able to experimentally validate and confirm what the flux control coefficients are for a given pathway of interest, meaning we can use these tools to tune up or down the expression of every single step along the way and see the impact that that has on the downstream part of the pathway. So the overall premise on this is if we can take a strong repressor linked together, in this case MXI1, linked together with dCas9 in a promoter in front of a gene of interest, we would have very, very low expression. It's going to repress it quite well. We found that as we step this element further and further away from the start site, we can actually begin to relieve that repression, meaning we get less repression. And then if we switch that over to an activator, we get activation of that gene expression. And as we step that back closer and closer to the overall transcription start site, we ultimately get higher and higher levels of actuation. So we can think about this as a knob in which we can use these elements to tune up and down the expression of various different genes of interest. And we showed that we can program this and multiplex it, meaning we can actually go through step in various different manners across a gene of interest, look at that change in the degree of actuation, and link that back to our phenotypic response, meaning we can do this mapping of change of gene expression to either an output of a pathway or a more global phenotype, such as a growth tolerance or a growth phenotype on a unique sugar, for example. So the tool really enables us to have this tunable up and down expression across different promoter elements. So here's just an example of where we took this psych promoter, which is a medium strength promoter, so about the first percentile or so in terms of expression. And by having this activator associated with it, we can get that promoter, depending on where we step along, to be as strong as that of a TEF promoter. We can also take an NDE1, which is a relatively strong promoter, and by stepping along a repressor along the site towards the start codon, we can ultimately repress that down to almost the level of a complete knockout in that regard. So these realization that we can create these broad spanning modulations of very strong overexpression to very strong underexpression is promising in that we can get the full dynamic range, and we can create these in a prefabricated type of manner, which allows us to really query pathways. So we wanted to do this in an iterative fashion where we can test for our phenotype of interest. We can use this STEPS approach to find a target of interest. Once we find a target that may be limiting our pathway, we can then eliminate that bottleneck by either doing a complete deletion, an overexpression, or some range in between. And we'll see why intermediate expression actually is important in this whole exercise. Nevertheless, we can create that next strain and then retransform that same set of guide RNAs to iterate this process to find the next N+1 target and iterate through to ultimately debottleneck pathways. So as an example on that, we studied glycerol production, something that had been known for quite a while within the field, especially on aerobic metabolism, the importance of things like GPD1 and GPP1 of needing to be overexpressed to improve glycerol production from glucose. Nevertheless, we took the genes that are really highlighted in bold, used the CRISPR-Cas9 type of approach to look at the impact of everything from strong knockdown to strong overexpression. And we can get these response curves like this. So here's an example of response curve associated with TPI1. We can see that for the most part, changing the expression lower and higher has no real impact. We do see a little bit of an increase in glycerol production as we begin to get to very strong knockdown conditions. And that makes sense in that the deletion of TPI1 would force half of the glucose carbon to go into DHEP and down into the glycerol pathway. Nevertheless, we did that for all of our other targets, and then we see one dominant target popping up. Obviously, a very strong improvement if we overexpress GPD1. And I note that GPP1, for example, is relatively flat across the board. But if we zoom in on the gene expression change patterning that we get with GPD1, we see that as we target this with our repressor and with our activator, we get strong improvements and increases in that glycerol titer, suggesting that GPD1 is a rate limiting step in the process, and therefore we would need to overexpress that. So when we introduce strong overexpression into a strain, into a background strain, and repeat the process now with the same exact guide RNAs that were sitting in the freezer, we get something that's a little bit different here. We notice that this TPI1, we no longer see that increase associated with strong repression, because now if we have a strong pull towards glycerol 3-phosphate, we don't want a deletion of TPI1. We actually want that reversible activity to be present. The rest of the targets are relatively neutral, with the exception now of GPP1 uniquely, has a increasing correlation as we go through and activate that gene, indicating that once again, this is the next rate limiting step of the process. So we can see that when we actually hard code wire these overexpressions in terms of our glycerol titer in grams per liter, going from our wild type strain to a GPD1 overexpression to a GPD1 GPP1 overexpression strain, we see this progressive improvement of our overall activity that we get. Likewise, we see that the targets that we identify are very specific to the genome, something obviously that's known, but we can see that relatively rapidly using these CRISPR approaches. GPP1 has no impact in the wild type background, but does improve the glycerol production in a GPD1 overexpression background. This really demonstrated the importance of being able to port these libraries into new cells of interest. We can do the same thing in another case study. So here we looked at DHS production, so looking at the aromatic amino acid biosynthetic pathway towards interesting both amino acids, but as well as other alternative products. We can deploy the same type of steps approach to each of the major enzymes in this pathway. We did that, and we see that across the board, focus more so on this area here, just because it's the one that's a little bit more relevant towards the targets that we ultimately identified, although ARO1 was an interesting overexpression target nonetheless. We saw that there was a strong suggestion of a knockdown or a knockout of ZWIF1, whereas it was relatively kind of ambiguous or flattened with respect to TKR1. So we can see, once again, strong improvement of our product down here of DHS if we had a strong repression of ZWIF1. So then if we were to delete ZWIF1 into our strain, redeploy the library to find our next new target of interest, we end up with relatively flat activity across the board, with the exception now of TKR1. When we zoom in here, now TKR1, so the transketolase enzyme, becomes a strong overexpression target as suggested through this approach. And likewise, as we hard code these into cells, we see the step from wild type with the ZWIF deletion and with the TKR1 overexpression in that background, really showcasing how this approach can rapidly prototype targets, but then also allow us to understand how to engineer the cell. Likewise, these targets are very specific to the genome. The TKL1 overexpression has only minimal impact in a wild type cell, where it has substantial impact in the background of a ZWIF1 deletion. So this really led us to the idea that we wanted to improve this dynamic range even further. And we, in subsequent work, were able to show that we can put a activator on DCAS9 and can do all the work that we did before, in those prior slides with just an activator and a Pol2-based promoter system, meaning we can use the activator to step along the promoter to get improvements in actuation. And we can also use this DCAS9 activator inside the gene as a steric hindrance type of approach and mechanism to be able to get repression. And if we target multiple sites, we get higher and higher repression. So we really can get over about a 20-fold range in expression modulation by using this type of approach, which is sufficient to be able to understand the impact of both strong repression as well as activation. We also made advances in terms of using tRNA-based systems to flank our guide RNAs so that we can get operons, if you will, of guide RNAs to express based off of a Pol2 basic promoter as opposed to using alternative promoter systems. So this allows for the cleavage and maturation of these guide RNAs and allows us to multiplex. Well, what that allows us to do is be able to multiplex rapidly pathways. So we looked at this for the production of 2,3-butadiol as an example. There had been some great work in the literature that suggested several targets that we would want to overexpress or delete. But now we can code that with guide RNAs and rapidly create a strain of interest. So we did that by designing 5 guide RNAs, 4 of which were repressive guide RNAs, so using our repressor of removing this ethanol production for the most part by repressing each activity. Now the alternative, what we did in the prior slides, now that the knockdown or repression of GPD1 to prevent the glycerol production, and a activator domain for the BDH1 to funnel more of our pathway towards this 2,3-butadiol heterologous pathway. So when we do that, we can actually recreate a strain very quickly. So we were able to recreate that engineered strain in a SynPK background and get about a 2-fold improvement of our titer, going from about 1.5 gram per liter to almost 3 gram per liter of production in that background. So that was able to show how we can rapidly strain engineer. Now the difference and challenge here is that this is something that we normally would have hard-coded and hardwired into the cell. But now we have the constructs. So we can simply put that into a new strain and test to see if there's a better background. So we took the same exact constructs and ported them over into a sigma strain. And that did pretty much the same thing. We all got a 2-fold improvement of our titer of BDO. But the key thing to look at is that these axes are very different. The baseline production of BDO when we put that pathway into the sigma strain is substantially higher, almost about over 5 grams per liter, which meant with that rewiring, same fold of rewiring, we're able to get almost over 10 grams per liter of 2,3-butadiol. So it really showcases the power of these CRISPR approaches to be portable rewiring systems, but then also, I think importantly, showcases the difference in diversity of yeast strains, which really opens even just within Saccharomyces, right? And one could imagine how this can be extended even further, but showcases the importance of looking at different background strains. So finally, I want to showcase a little bit of some of the newer stuff that we have just published in this past year, really expanding upon this approach in a much larger library-based approach that takes advantage of next-gen sequencing. And in particular, we looked at the stoichiometric model within yeast and wanted to create guide RNAs that stepped along all of the major stoichiometric-related genes. So we did this and used our design tools to create stepping guide RNAs, had a number of different types of requirements to be able to design these guide RNAs such that they would step to create alternative values for overexpression and repression, and then bin them into pools. So we pulled these into 4 pools, just mostly for convenience with our partners at the Joint Genome Institute as we're synthesizing these relatively large guide RNA libraries. And these were pools of carbohydrates and lipid metabolism, energy cofactors, amino acids, and nucleic acids, nucleotides, and other housekeeping type of metabolic function. So sort of 4 crude type of categorizations of metabolism, but really a ways in which to library-wise screen through targets of interest. So using these types of pools, we can, again, design these guide RNAs, express these with either the MXI or VPR linked DCAS9, transform these into cells, enriched somehow. And I'll talk a little bit about growth enrichment as well as product enrichment. And I know product enrichment is significantly enhanced as we move to biosensors, which I'm sure Jin will be talking about quite a bit in the next presentation. Nevertheless, either way, we can use then deep sequencing to look at the enrichment of guide RNAs that are suggestive of where we want to be in this overall patterning from strong deletion to strong overexpression. So we can use this as an example. We can use this overall tool. We use these MXI and VPR libraries, and we did growth enrichments in both galactose and glycerol independently, and then use next generation sequencing to be able to find where we found our enrichments of our optimal expression. So in doing that, we were able to kind of find clusters. So gene targets popped up in terms of their enrichment across the board. Some were in the strong knockdown category, some were in intermediate expression, and some were in the high overexpression, and then also intermediate overexpression. So we really saw that there was an optimum for gene expression for these targets. It wasn't just this binary, we need a strong overexpression, or we need a strong knockout to be able to get a phenotype. We often found our optimum somewhere in the middle. And we can see this with both the levels and targets that we get for both galactose and glycerol. So we can see that there are points of optimality where the gene expression was somewhere in the middle that we wanted. And then there's also points of optimality towards either the low end of knockdown or overexpression. But nevertheless, there was usually a peak. There was an optimum level of expression of a pathway enzyme for our targets. So just as an example of how this type of library approach can really uncover targets that are unreachable by traditional approaches, we're going to look a little bit at the glycerol library, for example. So here we found targets, in this case TPS2 and IPK1, that the knockdown was much better in terms of growth than the knockout. So the knockdown phenotype was better than the knockout phenotype. So if we use just a knockout collection, we would have missed some of these targets. There are some cases where intermediate overexpression was by far better than strong overexpression. So if we look at growth of here, the intermediate overexpression of GRS2, we get significant improvement of growth, but we get very little improvement of growth, actually just slight over the control in the strong overexpression case. And then we also found targets such as GPI-18 where we found an improvement with a knockdown, and the knockout is lethal. So essentially we're able to uncover targets that would have completely been missed through traditional gene knockout type of approaches. We also did some similar type of work with beta-xanthin. This is a product that is interesting because it's a pathway towards very interesting pharmaceutical agents, as well as beta-xanthins in general can be used as a colorant. So it allows us to do a very rapid color screen without having to need for more sophisticated type of biosensors in our screening system. So we used a plate-based screen to go through metabolism in this way, where we transformed our various different pools into our cells, looked at this on 96 well plates, screened for improvements of now secreted beta-xanthin, which was our key here for this work, and then used Sanger sequencing to look at our top pools and identify the guide RNAs that were most indicative and influential for this overall phenotype. So when we do that, we can see that using these 4 libraries, this is just the overexpression based constructs here, we can see that we get a range of targets, some of which, each of which the libraries have some improvements and some reductions of our overall fluorescence that we get, so essentially beta-xanthin production. We can see that in general, the amino acid and nucleotide pool was not as impactful as some of the other ones, including some of the housekeeping ones, as well as the carbohydrate metabolism and energy. So we get a little bit of in-depth information about the importance of our overall pools and the targets that are encoded in them. So, but at the end of the day, just like the example of glycerol and galactose, we're able to identify new targets that had not been identified before, and we can even multiplex them to get significant improvements in the secreted level of this product. But once again, notice there's improvements here, for example, of LAC1 and PDR11. These are important targets when they're, in this case, knocked down of expression, but a complete knockout removes that activity. So again, intermediate expression is critical in this type of approach. And we see that also with this target as WIF1 is kind of one that we went into a little bit more depth with. And here, just because we had these guide RNAs sitting in the freezer from the prior experiment, for example, looking at the DHS production, But we can see that as we step along here with improvements of repression, we see that it's pretty bad as we get really strong repression. But this intermediate level of repression is actually the optimum with respect to the fluorescence that we get, as well as maintaining constant growth rate. So we really showcase through this type of approach and these CRISPR approaches that this is a scalable technology that can be used and multiplexed relatively easily. We can see applications for pathway assembly and pathway optimization. We can get global optimization by doing that. And we can use these approaches to find targets, importantly, that would have been missed using conventional genetic approaches. So with that, I'd like to acknowledge the lab that really has done all the work and conducted all this research. In particular, the research that I showcased today was the work of 2 very talented graduate students and some undergraduate students and our collaborators at GGI, who provided some of the constructs that we saw here in terms of these guide RNA libraries. So with that, I'd like to thank everyone for their virtual attendance for this seminar, and I'll turn it back to John to introduce our next speaker, and then look forward to any questions that anyone has during the Q&A session. So thank you very much.
Thank you very much, Hal. That was great. I really enjoyed that. I think a lot of people are familiar with CRISPR for knocking out genes and doing mutagenesis. So you showed a whole new way of using CRISPR, and I can see already there's actually quite a few questions coming in. So don't go away, we will have questions for you. Yeah. I'm very pleased now to introduce our second speaker, Jin Hao from Shandong University in China. Jin, thanks very much for staying up so late, because it's nearly the middle of night for you out there in China. So I appreciate that, and very much looking forward to your talk. The floor is yours.
Thank you, John. I'm very glad to present here. Today, I will mainly talk about the biosensor design in yeast and the applications in metabolic engineering. I am in Shandong University, China. We are located in Shandong province. The university is located in Shandong province. It is in Qingdao. It's a very beautiful seaside city in China. So welcome to Qingdao when the situation is getting better. And we have the state key laboratory of microbiotechnology, which is one of the best microbiology lab in China. And we are aiming to bridge the microbial fundamental research into industrial. Sorry, I should put it back. In our group, we are developing tools for metabolic engineering and also for synthetic biology. We are also working on produce various products for industrial applications. As we know that the biosensor, which can sense the key metabolites in the metabolic pathway, has been used widely in metabolic engineering. For example, they can be used for high throughput screen, and if we couple the cell growth with the metabolites concentration, it can also be used for the continuous selection. And another application is the dynamic pathway regulation. And in general, there are 2 kinds of biosensors, including transcription factor-based biosensor or RNA-based biosensor. So here I will just give several examples of how to design the biosensor and how can we use it for the metabolic engineering. Right now, for the transcription factor-based biosensor, most of the sensor are found and built in the bacterial system. So if we want to use it in yeast, we have to transplant this bacterial system into yeast. In order to do that, we are using FEP-R, FEP-O system as an example to develop the design principle. FEP-O system is the system which can sense the change of melanoid, as we know that melanoid is a very important precursor, and it can be used for the flavonoid, the polyketide production, or the fatty acid derived chemicals. However, the intercellular flux in Saccharomyces cerevisiae is relatively low. Therefore, previously we have to use the transcriptional regulation to downregulate the phospholipid synthesis and improve the downstream product. The detection of melancholy is very difficult. That's why we need to build the sensors in yeast. The principle to build the activation regulation sensor is that we need to insert the FEP-R binding site FEP-O inside the a synthetic promoter of the co-element, and we insert it into the nearby the Tata box. And then through optimization of the position of FEP-O and the expression of FEP-R, we have developed a biosensor which has a relatively leaky expression and a very high dynamic output range. So here we developed the biosensor for activational regulation. Aside from activational regulation, we also design biosensor for repressive regulation. Different from the previous design, we have to insert the operator in the upstream region of the promoter, and then also fills the transferrin factor with the activation domain of the activator. In this case, when the melancholy concentration is low, it can activate downstream product. With the increase of the melancholy, the transferrin factor is dissociated from the promoter and deactivate the transcription. Therefore, the activation efficiency of the transcription factor is very important. Therefore, we evaluate several transcription factors, evaluate several activators, and found one activator, MED2, has a very strong activation efficiency. The efficiency is even higher than hydrode activator VPR. So in this case, we can build a very good biosensor with high dynamic output range. Aside from that, we also working on optimize the promoter to expand the dynamic output range. And if we want to decrease the basal expression, we can replace the co-elements into the synthetic co-element. For example, the minimal synthetic co-element, how just developed several years ago. So use this kind of design principle, we have developed both biosensor for repressive regulation and for activational regulation. Then we can use it for the metabolic engineering screen. Here we just screen the phosphorylation mutation of yeast ACC1 and obtain a mutant which can obtain a better titer of the melancholy downstream product. We develop a successful we have developed the biosensor successfully using FEP-O, FEP-R system. However, for the transcription factor-based biosensor, not all the prochromic transcription factor can be transferred into yeast system. So if in this case, can we still use those kind of sensor to screen yeast strain for the high productivity or high production? In order to answer this question, we construct another biosensor in E. coli system. We construct the glucanic acid biosensors. Until now, we still don't know exact machinery of this biosensor, but it has but the biosensor has successfully constructed in E. coli system, And then we put the biosensor into one stream and put the protein, the synthetic pathway into another stream. When we mix those strains into one pot, we see a very good response. Using this one pot, two strain system, we can screen the mutation library of the key enzyme in the metabolic pathway. And we also construct several yeast strains which can produce various titer of glucuronic acid. And we see a very good correlation between the fluorescence and the G titer. So in this case, even though we cannot transfer this biosensor into yeast system, we can still use those sensors have been built in E. coli and screen the yeast strain. Sometimes, even we have already developed the biosensor very successfully, perhaps it's still not good enough for the application in metabolic engineering. For example, several years ago, a student in our group has built another sensor. It's iron-based sensor. This sensor can sense the metabolite change of sialic acid. However, it can only sense up to around 4 grams per liter concentration of sialic acid. So in the initial metabolic pathway engineering, we can use this biosensor to optimize the metabolic pathway. However, right now the concentration, the titer is has already reached to around 10 grams per liter. It's already beyond the saturation concentration the aptamer can sense. So we have to we cannot use this sensor anymore. What can we do? So we do another round of, we did the direct evolution of this biosensor. We replace the GFP gene with another gene. It's a TET-A, which encodes a transporter. This transporter can transport the nickel iron into the cell or transport the tetracycline out of the cell. So in this case, when the medium has natural iron or tetracycline, the metabolite concentration is basically correlated with the cell growth. So we can couple the cell growth with metabolite concentration. Use this sensor, we did the directly evolution of the aptamer, and we screened the mutation library in a growth-based manner, and after several rounds of growth-based selection, we have obtained several mutations which can sense a higher amount of sialic acid and have a much better dynamic range. Use one of those sensors, we screen the RBS library of the metabolite pathway and also screen the mutation library of the key enzyme in this pathway. And combine these strategies together, the final titer of sialic acid has reached to 14 grams per liter, which is the highest when using glucose as the carbon source. So here we just demonstrate several examples of using the biosensor for high producer screen. Actually, aside from high producer screen, dynamic control of the metabolic pathway is also another very important applications. Compared to the bacterial system, actually yeast have relatively few examples on dynamic control of the metabolic pathway. That's because we are still lacking tools for dynamic control. Of course, we can use the tools of the biosensor, which can sense the key metabolite in the metabolic pathway to do the dynamic control. Aside from that, there's also another very important system. It's a chrome sensing system. Chrome sensing system can sense the change of the cell density and response and regulate the downstream genes transcription. Initially, we have expressed the bacterial Lux chrome sensing system into yeast, but it doesn't really work. So we have to borrow another system. This system is from plant hormone. So we construct the plant hormone cytokinin system into yeast and combine yeast endogenous YPD1 and SKN7 signal transduction pathways to construct the synthetic chrome sensing circuit. We have confirmed the location of the receptor and also expressed the enzyme which can synthesize the signal molecule. And then at last, we have proved that this system actually works in yeast, and it can sense the cell density and response in a density-dependent manner. However, we also found out there's a very high basic expression level of this system, and the dynamic output range is also not very good. So we want to optimize this system. We use protein engineering to do the optimization. We engineered the protein to control the promoter and to control the expression of GFP, or engineer promoter to control the production of the signal molecule. And then we indeed obtain several circulates which have a lower basal expression level and a higher dynamic output range. We also used some other strategies. For example, we optimized the expression of the protein in the signal transduction pathway, but it doesn't really work. So in this case, we choose this circulate and this circulate for the further dynamic control. Actually, in the metabolic pathway control, most of the time we want to downregulate the byproduct synthesis. However, it's a repression system, so we have to convert the activation system to the repression system. Of course, the first system coming into our mind is the CRISPR interference system, which Hao has already mentioned that. However, because the dynamic range is not really good in our system, we are not quite sure if it can work very well when we control Cas9 expression. So we have coming into our mind is another system. It's a protein degradation system. Protein degradation system is here we use the auxin-mediated protein degradation system. When we add oxytocin into the culture, the targeted protein can degrade it very fast. As we can see here, with oxytocin addition, the protein can be degraded within 100 minutes. Actually, we also found out even without oxytocin addition, we can still see protein degradation to some degree. And when we look into that, we found that actually yeast cell can produce oxygen and its precursor IAM. So that's why we can see the degree, the protein degree to some somehow. But when we express the enzyme which can catalyze IAM to oxygen, we can actually control the oxygen synthesis and then control the protein degradation rate. As we can see here, if we use a very strong constitutively expression of IAH, the protein degradation rate is very high. So in this case, we can use the chrome sensing system to control the expression of IAH. In this case, we can, actually we can control the protein degradation rate somehow. And use this system, we do the dynamic control of the metabolic pathway. The pathway we are choosing is the sucrine terpene production pathway, and we use this system to control the protein degradation of ERG9. ERG9 has been proved to be a very important protein to control the flux of sucrine terpene production. Our design principle is that when the cell density is low, ERG9 will not be fully degraded. So the metabolic flux can be directly to the scoline production and agosaryl production to maintain the cell growth. When the cell density is very high, ERG9 will be degraded, and then the flux will directly to the alpha farnesate production. Using this system, we found that alpha farnesate production actually increased around 80%. So it can work very well. And the production is much higher than constitutively degrade the ERG9. So here we just demonstrate a successful example to use the chrome sensing system to control the metabolic pathway. In summary, our study just demonstrated several examples and the design principle to transplant a prochronic transcription factor based biosensor into yeast. And we found out one pot two-strain system or directed evolution of the biosensor can facilitate the application in metabolic engineering. Although we have already designed the chrome sensing based dynamic control, actually more tools are still required to develop for the dynamic control of the metabolic pathway. So here is all on my topic. I want to say thank you to my collaborator, Professor Qinsheng Qi and Professor Juzheng Sheng, and I want to also want to thank my students in our group. I'm ready to take any questions. Thank you.
Jin, thank you very much. Really enjoy that, and I really like that concept that you were presenting at the end there, the idea that maybe density dependent switching on of pathways. I can see a lot of application for that. I'm about to go and look at the questions, but I'm going to buy myself a little bit of time by asking the same question to you both, and I'll let Hal answer it first to give Jin a chance to draw breath, and then maybe after Hal answers, if Jin could answer the same question. And my question really is, to what extent do you feel that the tools that you've described now, some or all of the tools that you've described now, are still at the stage or maybe are best applied for screening and maybe learning new biology around the pathways and regulation, versus to what extent are they close to being ready to actually use in commercial strains that would be used in a bioprocess? So I'll let Hal answer that, and then maybe if Jin could do it, and I'll be looking at the questions while you guys are doing that.
You know, I think that's a great question, and I think it's relevant for both of the technologies that we talked about. And I think there are certainly major applications and challenges associated with screening, no question, and the ability to either rapidly rewire a cell or rapidly control pathways and use biosensors to screen for improvements, I think is something that has really revolutionized the screening approach. And I think there's a lot more technologies that are required in screening of strains, as many groups and many industrial entities are looking at very large screening exercises that are mainly brought forth by the revolution of gene synthesis and rapid strain engineering. At the same time, a lot of these tools are sometimes still plasmid-borne. They have a lot of different parts associated with it, and in turn, they tend to be a little less stable, potentially long-term, than a hardwired, genomically encoded modification. And I think that's kind of what raises some of the challenges in using some of these types of approaches for further out industrial production. Second part is, I think a lot of the applications that have been done at the moment mostly because they've been done in laboratories, have been done on laboratory-based strains. And I think the importing of these tools, which is relatively straightforward but hasn't fully been done yet on more industrial yeast, is something that could be analyzed better to look at the stability of these various different type of constructs for ultimate deployment in industrial processes.
Yeah, I totally agree. Actually, I think for the biosensor, mostly currently we are still working on try to find some fundamental machinery underneath and try to find targets for further metabolic engineering. But actually, if, for example, if for the biosensor, if the concentration it can sense is very high, it's still possible to use it for industrial strains selection, of course.
Great. Thanks to both of you for those answers. So if people have questions, please type them in the question box on the right, and I will see them coming up on my screen here. Sorry if I'm leaning very close to you, but the writing is very small on my screen. There's a couple of people who have said thanks very much for the presentations, but I have a question here for Hal, and it's talking about multiplexing, and it says, what is the limit on different guide RNAs that can be expressed at the same time? So how many genes can be targeted at once? And then the follow-up is, what are the limits from preventing even more from being expressed simultaneously?
Yeah, so currently we have been able to express, I think the highest that we've tested is about 6 guide RNAs in succession on these tRNA, guide RNA, tRNA based type of constructs. I think the limitations here are twofold. One, just in terms of getting unique enough parts, so getting enough unique tRNAs and their validations, they don't impact the guide RNA function and folding. And secondly, just not oversaturating the system. Certainly, we have to have a balance between the amount of dCas9 that's available, because it's a multi-part reaction if you think about it, the binding of the DNA with the dCas9 and the guide RNA. So it is a little bit of competition, but I think the system can be pushed more. We didn't see necessarily any limits as we were expressing these 6. But it's just a matter of getting enough unique parts and balancing out the expression of these modulators for that.
Great, thanks, Hal. I have a question here for Jin, and it's talking about the TF-based biosensors, and it asks whether you can tune the limit of detection for the metabolite you're detecting. In other words, horizontal scaling of the response curves.
Sorry, my connection is not really good. Can you repeat it?
Yeah, the question is, can you modify the TF-based biosensor to tune the limit of detection for the metabolite you're detecting? And so it's
yes.
asking about horizontal scaling, so I guess going up and down in terms of the limit.
I think, yes, of course. I think it's possible to do the directly evolution to involve the biosensor, and then we can use it to for more metabolites, or to change the specificity, or change the dynamic range, change the threshold. It's possible.
Okay, thanks. And I have a question here for Hal. So it says, did you ever check the mRNA level of the proteins when targeting the genes of pathways? So did you test the RNA, and if not, did you ever look at whether the chromatin status was influencing the system? So I think it's asking, yeah, whether you've looked at expression by mRNAs, and then whether I suppose just positional effects on the chromatin and whether that's having an influence.
Yeah, so to answer the first part, yes, we do measure the mRNA. That's obviously the major mode of action of these dCas9 based systems is to change that expression level, and we have looked at that correlation between the mRNA and the output that we get, as well as the stepping along, and that's definitely has a strong correlation between those two. So we've done that for a number of different cases, and then have simply use that as a back validation after we go through and find targets. So we have found that the stepping approach tends to be quite reliable. As for the second part, there are cases where it tends to deviate sometimes a little bit, and chromatin structure does play, and like nucleosome positioning and whatnot does play a little bit of a role in terms of the design of these guide RNAs, but that can be dealt with a little bit more on the design side of the algorithm, and hasn't really influenced the overall net function that we get by the stepping along type of approach.
Okay, thanks. And just while you're there, Hal, there's a question. How strong is the bond between the guide RNA and the dCAS and the complex? Sorry, how strong is the bond between the gRNA dCAS complex and the target DNA? And is that tunable, thinking about industrial applications?
Yeah, so I think the in terms of the strength, I think there's a couple of things that can be looked at and discussed on that. I think at the end of the day, for implementation in an industrial strain, we want that net interaction, and it's not just the guide RNA and the dCAS, but also with the DNA, and then net the other factors, the other transcription factors that it's recruiting, right, to be a robust type of process. I think overexpression tends to be, an activation by those modes tends to be more robust than a repression tends to be. Reason being, if you have an activation type of scenario, even if there's a little bit of a loose interaction, as long as it's hitting that DNA, it recruits the factors, it starts making transcripts, you will get some sort of overexpression phenomena. In the repression mode, you require those elements, both the dCAS9 system as well as any other recruiting factors, to have to be there all the time. Because if they start to come off a little bit, then you can get errant expression at that stage. So I think the sensitivity tends to be more on the repression side than the activation side. So I think that's where, if there needs to be more work, it's on ways to enhance that type of repression. And so certainly we found, as I showcased on one of the slides, by having multiple sites that you can target at the same time during repression, you can get that to be a more robust type of signal by essentially removing that probability of having the element disassociate.
Thanks, Hal. There's a question for Jin as well. It's asking about the one pot, two strain system, and it's kind of a technical question, really, I think, but it's asking in the one pot, two strain, that this implies a kind of a co-culture of some sort. So could you just comment on, I suppose technically, how is that happening? Is it in a microtiter plate, or how does this work, Jin?
Actually, in those one pot two strain system, we just use the cell culture. For example, if we mix the yeast with E. coli together, we just use the yeast culture and then input it into the E. coli biosensor, so we can detect the fluorescence after that. So it's not a co-culture.
I see. Okay. And I'm looking at Hal. So there's a question there, Hal, whether it's possible to use your system in a di or tetraploid strains, or I guess triploid either. So yeah, polyploid strains, yeah.
Yeah. I think polyploid strains are interesting. We have certainly looked at some work more recently on expanding, kind of motivated by that work that I showcased, by moving from SEN-PK to sigma strain. Obviously, there are very big differences even in the haploid type of world there, but we have actually begun to look at different types of more wild yeast and polyploid yeast. I think the only challenge with these is understanding the sequence, right? Because the guide RNA is obviously sequence specific, so we have to know something about the genome sequence to be able to properly design those guide RNAs. Nevertheless, we have been able to see that we can port this system over to different ploidies on there, but at the end of the day, it really just depends on knowing the sequence of the genome.
Okay, and actually, I was wondering that myself. So in terms of porting the system, you kind of need to be porting it to a strain that's genotypically quite similar, I guess.
Yeah, if you were to use the exact same, meaning kind of the similar experiment that we had done with the butane dial, where we moved from sigma to rather from synPK to sigma, that was easy because the genome sequence is fairly similar. So the guide RNA would be transferable. Otherwise, the only thing that needs to be changed is that guide RNA sequence itself to make it organism specific. So if there is a very large deviation on the genome sequence in a different organism, in a different type of yeast, then you would just have to customize that for that particular yeast. The DCAS9 and the MXI or VPR and the like would likely work if otherwise.
Okay. And I have a question. I think this one is for you, Hal, but it seems to be inspired maybe by Jin's talk. But it says, could you discuss the use of sgRNA strategy in designing a general metabolic switch, changing the flux from growth to product formation based on the cell density?
I think it does merge the two presentations together quite well. And I think it requires obviously having control to be able to use these guide RNAs to turn on and off the profiles that you would want going from a growth phase to a production phase. But then it also requires sensing, right, to be able, through a biosensor, intracellular biosensor in this case, to be able to know when to activate that switch. And one thing that we've done, I didn't talk about this today, is that we've actually used T7 polymerase to drive the expression of our guide RNAs in some instances. And that's really unique because that's a very orthogonal type of control. So we can, and in doing so, we have showcased where we can actually then induce or turn on the expression of that T7 polymerase that then turns on the guide RNA. So you can almost imagine that type of sequencing where we can take that type of sequence where we can take some of the biosensors that Jin had just talked about and use that to not just drive the GFP or the TETs that she had used, but now to drive the T7 polymerase that then turns on the guide RNAs to start a cascade of metabolic rewiring. So I think such a thing is definitely possible by merging the type of technologies together, and could be very interesting applications there.
Yeah, you know, I should really have kept that question for last, but so just on the, this is one for you, Jin. It's a question about orthogonality, and because the systems that you were talking about, especially at the beginning, was a bacterial system going into yeast. Can you just comment on, I guess, the challenges and or benefits of using such orthogonal systems?
I think the challenge is that not all the bacterial system can be successfully transferred into a yeast system. But the good thing is that as long as we can create the biosensor, we can use it for the high producer screen. For example, I think perhaps when we combine those 1.2 strain system or combine some microfluidics technology together, it's possible to use the even in vitro biosensor to sense the metabolite change and to use it for the high throughput screen.
Okay, and actually there's another question just on the transplantation as well for you, Jin. Again, I guess it's a technical one, but the question is it better to integrate the biosensor into the genome or to have it on a plasmid?
I think it's better to have it on plasmid, and then we can remove it anytime if we want.
Okay, right, but and that's a selection pressure, but obviously you just maintain the plasmid then with your marker, I guess. Yeah, and somebody here, I don't want to be giving names, but somebody who was very impressed with your talk, Hal, and wants to know, is there any database where we can get the information for expression, activation, and repression for the extensive list of promoters already characterized by your group?
Yeah, so actually in our more recent paper that we had published in PNAS just this past year, in our supplemental information, we have kind of one of the supplemental information sections has a Excel file that essentially has the various different expression levels that we have from the deep sequencing that we measured, as well as the sequences for those guide RNAs. So I think that would be a good resource there for people to look at.
Yeah, absolutely, and I know there's lots of other resources from your lab available in different places as well.
Sure.
Okay, everybody, I think we've gone for about an hour and 10 minutes here, but I think the discussion has been very interesting. I'd like to thank the people who asked questions, because I thought we had some very good questions that came in there. I think I covered everyone's questions. Apologies if I didn't. If I missed your question, maybe you could just email Hal or email Jin and ask them directly yourself. I'm sure they'll be happy to respond to you. I'll thank, just in the background, Sarah and the people from OUP and FEMS who helped set this up and are running the webinars. Hal and Jin, you've set the bar very high, I think, for the future webinars in this series. So thank you very much. We're going to have another webinar in January on to do with yeast genomes, and I will be telling our speakers there that they've a hard act to follow. So thank you very much, both of you, for those presentations. I think you showed really that the technology, it's advancing so fast, it's amazing. And I think that the concepts of integrating the two aspects of what what both of you guys talked about to develop the next generation of metabolically engineered strains for biotechnology, it's kind of exciting. So really enjoyed it. Thank you very much. And we'll sign out.
Yeah, thank you very much for the invitation and for all the attendees. And we're more than happy to take any questions if people have that via email. So certainly.
Thank you very much, Hal, and then thank you very much, John. I really enjoyed it, so I'm also happy to take any questions.
Great. Thanks, everyone.